Hi everyone, in this recording, we will study a bacteriophage called as FX174. This bacteriophage FX174 infects E. coli. E. coli phage FX174 contains one circular single-stranded DNA molecule. So far we have learned T4 bacteriophage, lambda phage, T7 bacteriophage, all having double-stranded DNA as their genetic material. Now, this phage is a single-stranded DNA phage. It has single-stranded DNA as its genetic material, and the single-stranded DNA is circular, consisting of 5,386 nucleotides. The base sequence is known for this FX174 DNA. DNA is enclosed in an icosahedral head composed of 3 coat proteins and 1 internal protein. The phage has only 11 genes. So the genome of this FX174 bacteriophage is very small, just 5,386 nucleotides. So with such a small genome, it's even difficult for the virus to have 11 genes or to code for 11 proteins because the genome size is so small, but the phage still has 11 genes because the phage has managed to have overlapping genes. The number of amino acids contained in these proteins, 11 proteins, exceeds the coding capacity of this very small DNA molecule, but FX174 has efficiently evolved to have overlapping genes that are translated in different reading frames. So you can see here, this is gene A. It starts from here and ends here. And then the gene A star is within gene A. The gene A star starts here and ends somewhere at the same point. Then the gene B also is within gene A. It's very small gene, so the gene B starts here and ends here. The gene K starts near the end of gene A and runs into gene C. Gene E is also within the gene D. These are the 11 genes and these are their functions. The gene A codes for the A protein, which is involved in replication and viral strand synthesis, which we'll see later. A star is involved in turning off of host DNA synthesis. B protein and D protein are involved in the capsid formation. C is involved in the formation of phage unit-sized DNA. E is involved in lysis of the host. F is the major coat protein. G and H are major and minor spike proteins respectively. H is also involved in the adsorption to the host cell, which we'll see. J is a core protein and the function of K is unknown. Phage FX174 adsorbs to its host E. coli by means of a spike protein. When we talk about bacteriophages, most of the times we talk that a bacteriophage attaches to its host bacteria via tail fibers and the pins, and the hexagonal base plate settles down, and then the DNA is injected inside the host. And the rest of the proteins of the phage remains outside. This is what we have learned in case of T phages and other phages. But in contrast to T phages, here along with DNA, a protein also enters the bacterium. So the terminal spike protein coded by the gene H. So this H protein, which is a minor spike protein and which is also involved in adsorption to the host cell, this terminal spike protein or the H protein enters the bacterium with the DNA. So this point here, the FX174 bacteriophage differs from T phages or other phages. T phages do not inject proteins, but here you can see that FX174 is injecting a protein as well along with the single-stranded circular DNA. This H protein is required in an unknown way for converting the bacterium to a phage factory. Also, when we talk about bacteriophages, we say that the protein generally never enters the host, only DNA is injected, and we say DNA is injected into the bacterium. like a syringe and a needle, like how T phages do. But the DNA of FX174 is not injected into the bacterium. Instead, it is released into the cell surface, and from there it's taken inside. This is known because shortly after adsorption of the FX174 phage particle to a bacterium, the phage DNA can be digested if DNase is added to the suspending medium. Such DNase enzyme sensitivity is not true of any of the tailed phages which inject their DNA into the bacteria. Removal of the coat protein is coupled to replication of the parental DNA. Now we'll see the replication of FX174. So this is the FX174 phage with the icosahedral head. This phage infects E. coli. It adsorbs to the host via an attachment protein. The DNA is released on the surface from which it's taken inside. The H protein enters along with the DNA in the cell. The genetic material of FX174 is single-stranded DNA, which I've already mentioned, circular. So you can see here circular single-stranded DNA, and it's the plus strand. After the uncoating happens, the replication or the mode of the replication of FX174 is especially interesting or very interesting because the phage has the problem of making an identical copy from the single strand. The genetic material of this FX174 is single-stranded DNA, unlike your T phages or lambda phage. So the phage has a problem of making identical copy of this single strand. The template cannot yield progeny DNA molecules in a single step, or the replication cannot take place in a single step. So what happens? So first, when this DNA is uncoated, this DNA is coated with SSB protein, single strand DNA binding proteins inside the host. And a complementary strand has to be made. This occurs before transcription begins, and it depends on the host enzymes exclusively. This complementary strand that would be made from this plus strand or the parent strand of the virus, the complementary strand will be made before transcription, and it depends on the host enzymes. Many enzymes are involved, host enzymes are involved here, primase, polymerase, ligase. And when the complementary strand will be made, this parental strand of the virus is a plus strand, so the complementary strand would be the minus strand. Once the minus strand is made, I can say that the single-stranded DNA molecule of the virus or the plus strand is converted into a covalently closed double-stranded molecule, which is called as RF1. This is one, replicative form 1. This is an intermediate, you can say, or replicative form 1. This is supercoiled also, and the SSB proteins are removed when the complementary strand is made. The complementary strand, which is the minus strand, that is the only strand that is transcribed, that is the coding strand. So minus strand se abhi transcription hoga. So that is another thing you can say when this phage infects E. coli, and when the uncoating happens, and when it's single-stranded, replication cannot proceed in one step. Yeah, that's true. But also the transcription cannot be done unless the complementary minus strand is made, because the complementary minus strand is the coding strand or the strand that is transcribed. Once the RF1, the double-stranded covalently closed circle is formed, transcription would take place, mRNA would be made, the mRNA would be translated into protein, and a multifunctional protein is formed, which is the A protein. This A protein, which you can see green in color in this picture, A protein will come and make a single strand break or nick in the plus strand. So the A protein will make a nick here. In which strand? In the original plus strand of the virus. It will make a single strand nick in the plus strand between bases 4305 and 4306, because that is the origin of replication. This A protein, after making a nick, doesn't leave it remains there covalently attached to the 5' phosphate terminus, and this nicked molecule with the A protein linked is called as RF2, that is the replicative form 2, or ARF2 complex. That means the A protein is linked to this replicative form 2 now. E. coli proteins, which are synthesized prior to the infection, will cause the parental plus strand to be displaced from the replicative form 2. So the A protein, which is the protein of the phage, has just made a nick. It will just make a nick. It can't do the entire replication. So the E. coli or the host proteins which have been synthesized prior to the infection will cause this nick plus strand to be displaced from the RF2 by loop rolling circle replication. Yes, the FIX174 bacteriophage replicates via rolling circle replication. As we know, viruses, some viruses do replicate via rolling circle replication mode or the sigma mode. And when this 5' for the plus strand would be displaced from the RF2, meanwhile, the synthesis will continue in the circle. And it's not like that when you pull apart the plus strand completely, again वो single stranded बन जाएगा. No, the synthesis continues in the circle. This plus strand, when it's completely displaced and when it completes the one round circle, it will be displaced, it would be cleaved, recircularized, and you would say what happens to this plus strand which is cleaved and recircularized? Again, it goes here and the complementary minus strand would be made, resulting in more RF1. So there would be synthesis of many copies of RF1. Is there a time when this plus strands, rather than going for the synthesis of more RF1, will be packed into the capsid? There is no switch from synthesis of RF1 to the synthesis of free plus strands. Instead, the packaging system, that is the head icosahedral capsid, captures these plus strands before they can serve as the template for the synthesis of the minus strand, or before they can go for the synthesis of RF1. तो माइनस स्ट्रैंड कॉम्प्लीमेंट्री बनने से पहले और RF1 बनने से पहले दिस प्लस स्ट्रैंड्स व्हेन दे आर क्लीव्ड एंड रीसर्कुलराइज्ड, द पैकेजिंग सिस्टम और द आइकोसाहेड्रल कैप्सिड और द हेड कैप्चर्स देम एंड पैक देम. This capturing is delayed until completed phage heads have been synthesized. Now here when I said RF1 बनने के बाद ही transcription होगा and translation hoga, I spoke about A protein, but the other proteins would be made. The head proteins, the spike proteins would be made. So unless the phage heads have been synthesized, how will they capture? So this capturing is delayed until completed phage heads have been synthesized and begins when there are 30 to 40 copies of RF1. At that point, most of the DNA is engaged in loop rolling circle replication, producing new RF1 and some plus strands for packaging. Host DNA synthesis finally stops at this time. When all these things are happening, the E. coli replication or E. coli DNA synthesis finally stops as a result of an unknown function of A protein, and partly because all the host polymerase is engaged in the synthesis of FIX174 DNA. Because with the exception of the A protein, which you can see here, all proteins used in the FIX174 replication are the host proteins or derived from the E. coli genes. As more heads are made, all these plus strands are packaged into the heads and synthesis of replicative form 1 stops for the lack of template. This is another picture or another diagram of the same process is shown in a little more colored way. So you can see here the FIX174 attaches and enters, and the single stranded DNA, which is circular, is released. It's coated with SSBs. Then the complementary strand is made. The parental plus strand is shown with dark blue color. Complementary minus strand is shown with the sky blue color. Complementary minus strand is made, resulting in the formation of double stranded DNA, covalently closed DNA, that is replicative form 1. After this replicative form 1, transcription would take place. This mRNA would be translated. One of the proteins would be the A protein. A protein would go and make a nick here between the 4305 and 4306 base in the plus strand, that is the origin of replication. Once A protein has made a nick, the plus strand would be displaced by the host enzymes, and you can see the plus strand is being displaced and the synthesis continues in the circle here. And when this plus strand completes the circle, the plus strand would be displaced, it would be cleaved, it would be recircularized, but you can see वो plus strand निकलने से the circle has not become a single stranded. The synthesis is continued in the circle, so the circle is double stranded, but the plus strand has completed one circle. In this step, it is cleaved. This is loop rolling circle replication, and the plus strand is circularized, and it again goes for the synthesis of the complementary minus strand, resulting in more RF1. And when heads are made, these plus strands will be captured for packaging into the heads when the heads are made. And before they can be sent for the synthesis of RF1, they are packed, and that's called as encapsulation. So the replication basically has three steps: conversion of the parental plus single-stranded DNA to a covalently closed double-stranded molecule called replicative form 1, synthesis of many copies of RF1 via loop rolling circle replication, and synthesis of plus strands for encapsulation. Now, transcription of φX174 DNA. All transcription is from the minus strand. So transcription cannot occur until the minus strand is made. And from then on, the RF1 synthesis continues unabated until a sufficient number of heads are formed to start packaging. φX174 DNA molecule is very small, and many rounds of replication are completed before the head proteins are synthesized and assembled. The first completed particle appears about 10 minutes after infection. Lysozyme activity is delayed because translation for that enzyme is slow and the enzyme is not particularly active. Lysis does not occur until 30 minutes after infection, by which time about 500 Phage particles have been synthesized. We move on to, once the replication is over, the transcription is over, the heads have made, how packaging of the φX174 DNA takes place. Packaging of φX174 DNA requires 7 Phage proteins, and only 4 of them are present in the final finished particle. We will be studying the 7 steps of the packaging here, taking each step at a time in detail. So the first step is the head protein, which is the protein coded by the F gene. Okay, the head protein coded by the F gene or the F protein, and the spike protein coded by the gene G, that is the G protein. This head protein and spike protein form pentamers. You can see here 5F and 5G. So 5 copies of F protein and G protein associate, they form pentamers, and in the presence of one more protein called as the B protein. The B protein is also joining F and G, and the F 5 copies and the G 5 copies along with the B form a structure called as the 12S particle or the 12S unit. Next step, H also are added to complete the spike. The H protein or the protein coded by the H gene are added to complete the spike, which you can see here. The H also has been added. And the D protein forms a frame or a scaffold on which a prohead is built. During the packaging of any bacteriophage, directly the head is not formed. First, a prohead is formed, which is immature head or not the final one. That's why it's called as prohead. And from the prohead, the mature head is formed. So the D protein, which is coming into the picture here, forms a frame or a scaffold on which the prohead is built from the 12S units. Thus, the prohead, which you can see that this structure contains B, D, F, G, and H. F and G are the head protein and the spike protein, which have formed pentamers in the beginning, and B protein also were added to that to form the 12S structure. After that, H was added, and then the D provided a scaffold or a frame on which the prohead was built. What next? Then the C protein, binding to the 5' end of the displaced plus strand, somehow directs the 5' end into the prohead. So there is a protein called as C protein, which you can see here. And this C protein is binding to the 5' end of the displaced plus strand, which is being, cleaved from that loop rolling circle replication process. So C protein binds to this 5' end of the displaced plus strand, and it just directs the 5' end into the prohead. C is not the final protein. C just shows the DNA way to enter into the prohead. And then finally, there is a J protein also, and this J protein you can see organizes the DNA inside the head, circular single-stranded DNA. Next, fifth step, D protein, which was added at this step to F and G to form the 12S unit. The B is released from the prohead because B is not present in the finished particle. The A protein, which is involved in creating that nick in the plus strand between 4305 and 4306 at the origin, and it also stops the host DNA synthesis with an unknown function. It also involved in the packaging where it cuts the displaced plus strand from the loop rolling circle and circularizes the displaced strand. The D protein, which provided a scaffold or a frame on which the prohead was built, the D is also removed, leading to the formation of the finished Phage particle. So in this 7 packaging steps, whatever proteins you can see highlighted with blue and pink are not present in the finished particles. The only proteins present in the finished particles are the ones shown in the red, that is F, G, H, and J. The finished mature Phage have F, G, H, and J, and D protein, which provided scaffold, is removed. B protein is released, which helps in formation of the 12S unit. C just directs the DNA into the prohead. So, and A just makes a cut. So the finished mature particle only has F, G, H, and J. So this was the packaging of φX174 bacteriophage. In this video, we learned the properties of φX174, how its genome is very small, how it has still 11 genes and codes for 11 proteins because it has overlapping genes. Some distinguishing features or the hallmark of φX174, like the H protein enters along with the DNA, it's not injected, the DNA is released on the surface, then the replication, transcription, and finally the packaging of the DNA of φX174.